question being presented in this research study is, what is the origin of causation for structurally defective centrioles found in tumors? And the experimenters formulated a specific hypothesis, and because it is known that the overelongation of centrioles could be associated with mitotic polo-like kinase, or PLK1, which is responsible for the regulation of centriole elongation in human cells, the experimenters predicted that abnormal PLK1 levels could be responsible for the defective centrioles produced in these tumors. So the experiment that researchers designed was to observe the effects of various lab-induced disruptions to different phases of mitosis that were specifically targeted as phases in which elongation typically occurred. And to look at how different disruptions that were produced in the lab to these different phases of mitosis affected the process of centriole elongation. In order to do this, they first obviously identified where in the cell cycle elongation most commonly occurred using EM analysis. And then they utilized MLMC-D3 cells and RPE1 cells to actually visualize how these defects impact ciliation. And to do all of this, they first created a cell culture and synchronized the cells using a specific drug treatment and mitotic shakeoff. And then they utilized immunostaining and immunolabeling in order to actually visualize centriole elongation at different phases in mitosis. And they used wide field microscopy and structural illumination microscopy to observe the actual centriole elongation in a given moment, and then followed by storm imaging and EM analysis. And then the centrioles were measured, and the resulting data and imaging produced from all of these methods was statistically analyzed for significance.
What is the origin of causation for the structurally defective centrioles found in tumors caused the researchers to turn their attention towards PLK1, which has shown to associate with the centriole elongation factor CPAP. What researchers ended up finding out was that PLK1 plays a dual role in the centriole cycle. Researchers were also able to find out that PLK1 overexpression and inhibition can influence procentriole elongation. Researchers were also able to provide an explanation and proof of why overactive PLK1 is frequently associated with centriole structural abnormalities. The researchers were also able to provide a demonstration from their data of why numerical and structural centriole abnormalities often coincide with tumors. What ended up happening and new information found out was that PLK1 has previously been shown to promote the biochemical maturation of centrioles. PLK1 is also able to stimulate elongation of procentrioles, while at the same time promotes their gradual maturation, which ultimately in the end reduces their ability to elongate. What researchers ended up also finding out was that PLK1 could represent an effective way to couple these two processes and asserting some consistency in centriole size during cell cycles.
So finally, I did the conclusion. And basically the question we're asking is, what biological conclusions can we draw from this experiment? First of all, human cycling cells don't have a set structure for monitoring centriole elongation, which means that there's no one mechanism that ensures that centrioles are formed at an exact length, meaning that there are many factors involved. This causes the elongation process of centrioles to vary throughout the cell, which is what the researchers were primarily studying. Researchers predicted that these varying elongation patterns could signify that elongation factors vary in concentration in different locations within the cell. Researchers were able to speculate that any event that causes mitotic delay may potentially promote centriole overelongation, providing a cause for this phenomenon. Below, I have a quote from the actual study, which states centrosome numerical errors can originate from aborted mitosis, from cell fusion, or from centriole amplification. Our data imply that all of these events can promote the formation of centriole structural aberrations. This can explain the origin of aberrantly structured centrioles in tumors and why centriolar structural defects are frequently found in tumor cells, along with numerical defects. And basically, to conclude, this study and its results explain the origin of centriole variability in cells and why that and other centriole abnormalities are associated with overactive PLK1. This demonstrates why centriole abnormalities are often found in tumors.
I researched what is next, so these are some things that came to my mind after reading the article, or some things mentioned in the article by researchers that were still unclear, and these are things they could do to continue this study. One thing is to determine if PLK1 hyperactivity has any other effects on centrioles other than just overelongation and altering the centriole structure. What other cell structures could PLK1 hyperactivity have effects on other than centrioles? How PLK1 actually regulates procentriole elongation, examining exactly why these centrioles lack a mechanism that monitors elongation, and kind of going along with that point, determining a means of incorporating an elongation monitoring mechanism into these centrioles in a lab setting of some sort, and then studying these effects of having such a mechanism. What does this do to help the centrioles? Because this overelongation is a result of the lack of an elongation monitoring mechanism. So having this mechanism, how would this impact the centriole as a whole, since it's not able to overelongate? Discovering the extent to which cells can tolerate variations in centriole length without harmful consequences. So basically, how long can these centrioles get without it having a negative impact on the cell as a whole? Whether and to what extent PLK1's functions in procentriole assembly overlap with those of other polokinases. And then finally, determining why the percentage of cells that form overelongated and structurally inconsistent procentrioles, why does this vary among cell lines? So these are just a few ideas that researchers could use to continue this study.